Thank you everyone for joining today's Fireside Chat with the Salk Institute. I'd like to welcome Talmo Pereira and Jeff Denworth. Welcome, Talmo.
Thank you.
And Talmo's a Salk Fellow and Principal Investigator at the Salk Institute, and we're going to hear more today about the work that he does with the Salk Institute. And Jeff, if you want to go ahead and introduce yourself.
Sure. I'm Jeff, co-founder of VAST.
All righty, awesome. Well, excited to get into it. Jeff, I will hand it over to you as we get started.
Okay, Talmo has like a billion slides to go through, okay. So I think maybe a little bit of origin story here. We did a fireside chat with another customer at Nurse years ago, and actually it was about a year and a half ago now. And it was just like such a nice conversation where you get to explore how people that aren't exclusively steeped in storage think. And so as I travel the world, I kind of think, okay, well, who else would be kind of like an interesting personality for these types of discussions? And when I first met Talmo, I saw that same spark where I thought, okay, we could put together a really good conversation. So the Salk Institute probably is not a household name brand all around the world, but Johannes Salk is credited for basically curing polio. I read a statistic this morning where there was 45,000 cases of polio in the U.S. before the vaccine was discovered, and then the following year there were 910. And Salk never charged a single dollar for it. The whole world benefited. And now the world continues to benefit from the research that's conducted by the institute that bears his name, that's located in La Jolla. Is that right, Talmo? Talmo's calling in from Texas, where he's at a NASA conference today. And he was really kind of gracious with his time and offered to join us. So that's kind of the quick background on Salk, and you can infer from this discussion that they're also a VAST user, and we'll talk a little bit about that. But the floor is Talmo's, and he's prepared some kind of like introductory discussion slideware just to kind of help you all understand the research that Salk is conducting and that he's responsible for and involved with. So Talmo, take it away. The floor is yours. Show us the world through your eyes, and then we'll have a conversation.
Awesome. Thanks, Jeff. Thanks, Boone and Chuck, for putting this together and for giving me a little bit of time to share some of the work that we're doing here in my lab and at Salk more broadly. So the title for today is Quantifying Behavior and More using Deep Learning. And this is the work that my lab does here at the Salk, or there at the Salk. To start, let's just take a look at a little bit of nature. Let's motivate a little bit of some of the cool AI work that we're going to be doing. And it all starts with the very fundamental observation that all forms of life move, right? Whether it's a plant growing through soil, a fruit fly grooming its head, that's what's on the second panel there, or even a giraffe walking through a savanna. All forms of life evolve the ability to move because movement is important for survival. You need to move away from threats, move towards food, move towards mate. So it's something that evolution really hard-coded into biology. It's so essential, it's such an important function that evolution even gave rise to the nervous system and eventually brains, just so that you could move. So given its universality and its central importance, wouldn't it be great to have a good way to quantify it? But in other words, wouldn't it be great to be able to go from images that look like this to the distilled out core elements and degrees of freedom that might look a little bit like this? So what we're going to be talking about over the next 10, 15 minutes is going to be first some of the work that my lab does in motion capture using deep learning. And then we'll end with just a short vignette on how we've been applying this to better understand and diagnose disease. Cool. So starting with this first section here, we just talked about all these different kinds of movements. And the specific task within computer vision that boils down to essentially motion capture is going from images like this to track the location of individual body parts that look like this. In essence, it's the equivalent of putting a motion capture suit on a plant, on a fly, on a giraffe, without the suit. So when we first started working on this problem, we looked around and we saw that there existed methods for human pose estimation that were developed in the field of computer science for, again, tracking person movement. And this really relied on having massive data sets of individual humans that were already annotated at great expense to lots of industry labs that could crowdsource this annotation and a construction of these data sets of hundreds of thousands of images. The problem is, anytime you're in a lab setting and you have a new animal, a new experimental condition, a new camera, a new illumination, none of those data are going to help you at all. It's a different body, it's a different type of imagery, image statistics, and so forth. So your deep learning system is not going to generalize to these new settings. And so in my PhD, one of the things that I worked on was in developing a system called LEAP. LEAP stands for Leap Estimates Animal Pose. It's a cute double recursive acronym by my then undergraduate mentee, Diego Altarondo. And it works in a similar way to the human pose estimation models. It takes a raw image as input, here a fruit fly. It feeds it into a convolutional neural network. Neural networks are essentially the backbone of modern AI. And the neural networks learn to predict a representation of the location of those body parts. Essentially, these are heat maps, or effectively a spatial representation that represents the probability of each body part being located in each part of the image. Our key insight was to engineer an artificial neural network that was sufficiently lightweight such that it could scale down to the sizes of the data sets that were available for these scientific applications. We applied it to all sorts of things, and it just worked right out of the box. And I'm, of course, hiding away, summarizing, I was skipping over lots of work that was done by lots of amazing people, but suffice to say that we worked out, engineered a solution that would work well, even in this small data set. But one of the domains that we were particularly interested in was in applying this to the domain of multiple animals, where while it was trivial to extend our systems to do pose estimation on single animals to multiple animals, in the sense that we could locate multiple heads and multiple tails and so forth, it was a much harder problem that we had to solve to figure out which head goes with which tail, right? It's not something as simple as whichever one's closest. It's especially not the case when animals are closely interacting and they're essentially all intertwined with one another. So this requires reasoning about the entire body and not just body parts. And then finally, this problem of identity assignment, which is essentially now reasoning not just about what a body is, but really what an identity is. What is the same animal across multiple frames in time? And this now corresponds to both being able to reason about body parts, bodies, identities, and movement. All put together, serves for a very high-level task. So to address these technical challenges and recognizing there was not going to be a single solution to fit all, we developed a successor of LEAP called SLEEP, or Social Leap Estimates Animal Poses. And it was designed from the ground up as an end-to-end framework that takes you from having raw videos through to doing the data annotation, through to training or configuring training your neural network, applying it to new data, and pulling it back in for correction. But in other words, it marshals the power of deep learning to go from raw videos through to the creation of models that can automate the labeling of any number of body parts with any body configuration and any number of animals in any species in any experimental condition. So suffice to say, generality was really the name of the game here. And so one of the aspects of SLEEP that our users particularly appreciate is its ease of use, right? So it's something that's as simple as clicking and dragging. You drag and drop your video, define the body parts you want to track, create a little queue of images that you're going to label collaboratively with the AI. And then from there, it is just point and click, right? You're going to click and drag these body parts. You can copy paste them. You can rotate them. And you're essentially going to do this on a handful of frames, not 250,000, but something a little bit closer to 20 to 50, right? And so in this little sped up demo, what we're going to do is we're going to label about 5 images, very riveting. We're going to configure our neural network here, and then immediately it starts to train, and you can essentially see what the neural networks are thinking, right? We're outputting in real time the predictions of the neural network in terms of where it thinks body parts are located, from where we can then essentially then import the predictions and further correct them, essentially closing the loop between AI and human annotation. Cool. So really, this whole software is only enabled by a very talented team of software engineers, and essentially an industry-grade system where after I spent a little bit of time working at Google, I really learned what it takes to create a production-quality, industry-grade machine learning system. And that's what is represented in this little diagram here on the left. Every one of these little boxes has a story, and we'll talk a little bit more about it. But suffice to say that unlike lots of scientific software, our emphasis on accessibility and proper engineering really has allowed this tool to take off, where we now have thousands and thousands of users all around the world who have used this software to study all sorts of behaviors in all sorts of animals and all sorts of biological systems. And it continues to grow. All right, so because I want to keep this tight, I'll just mention one of the little stories that we have here along in this whole system. Like I said, each one of these boxes in the sleep framework has its own story associated with both the engineering advances and what kind of new science it enabled. But I'll just show you a very, very abridged version of one of these, which is a capability that enabled us to create what we like to call social mind control. Essentially, after working with some folks at NVIDIA to integrate TensorRT, a technology that can speed up inference of the types of neural networks that sleep can train, we were able to get down to just 3 milliseconds inference latency. What that means is that within 3 milliseconds, we can grab an image from the camera and predict the locations of all the body parts and track all the animals within it. And we use this with a couple of fruit flies. And specifically, this is a male and a female fruit fly that are engaged in their courtship ritual. Fruit flies court by essentially having the male sing to the female by extending his wing and vibrating it, and then approaching her once she's sufficiently sexually receptive. And by design, we had made the female sexually receptive in a way where if the male were to approach her, they begin to copulate. But because we can detect that they're doing this little song and dance in real time, one thing we can do is we can optogenetically activate. Essentially, we can activate the neurons in a female fly's brain, causing her to artificially reject him in a situation where she would not have otherwise. And this is something that we can do reproducibly. And all enabled by essentially the engineering advances that were created as part of this framework. Okay. One of the new directions that we're going in with this technology is to essentially enable 3D pose tracking. This is a collaboration with some folks at Princeton, in which we're now extending sleep to work with having multiple camera views, like you can see here on the left. And we've developed new approaches that allow us to reconstruct the 3D location of all of the individual body parts of these animals, and soon also their environments. So we can kind of tell exactly what they're doing, even when they're doing things that aren't within just constrained to a two-dimensional plane. We're also using this in other settings, such as with humans, where here you can see what we've done is we've reconstructed part of a museum environment in the LA County Museum of Art. This is a collaboration with the LACMA and Talmo and Sergey Gepstein at Salk, in which we're applying our 3D motion capture technology to track multiple individuals, place them within this virtual reconstruction of this space, and then be able to do things like retarget the camera so we can reconstruct what people are seeing. And incidentally, this is a massive data set that we're in the process of analyzing that has on the order of over 100,000 museum visitors whose trajectories we all have to trace along by extracting it from tons and tons of video footage. Okay, and one last application before we move on to the disease stuff is climate change. And so when I joined Salk, I joined the Harnessing Plants Initiative, where this is a collaboration between several labs at Salk, whose main objective is to phenotype plants by essentially trying to characterize their root system architecture properties. Essentially, what we want to do is we want to develop plants that are that have deeper roots, more massive roots, and roots that produce more carbon-rich polymers, such as subrin, or effectively cork, all processes that can be upregulated biologically or genetically or through natural variation, but that have the consequence of sequestering more carbon. And this is roughly what it looks like when we apply sleep to it, which then we can use to extract all sorts of traits like the tip depths, the rooting angles, and all sorts of other aspects of their geometry, which then allows us to basically do more machine learning to classify whether a particular plant has more or less carbon sequestration capabilities. This allows us to then screen through lots of different variants of common crop species, for example, which serve as excellent targets for enhanced carbon sequestration. Okay, that's this first part here, all about motion capture. And in these last couple of minutes here, I just want to quickly give you an overview of or a quick glimpse of our disease phenotyping efforts. And so one natural application of sleep and motion, deep learning-based motion capture is just to do it at scale, essentially. And so to do this in collaboration with lots and lots of folks here at Salk and beyond, we've been developing a large-scale phenotyping system, of which VAST is an integral part. It's essentially a system that allows us to capture the behavior of our preclinical rodent models. Essentially, we have mice that live in their home cage. It's a little cage at the bottom of these boxes here, from which we can basically record video 24/7 at 50 frames per second at high resolution, and basically be able to capture all of their movements, from which we can then derive phenotypes like tremors or motor coordination and so forth, that can then give us insights into different kinds of diseases. And we're specifically targeting amyotrophic lateral sclerosis, Alzheimer's disease, pancreatic cancer, and a few other neurodegenerative and psychiatric diseases, all through the same technology and the same platform. Now, of course, the challenge with doing 24/7 recording at high resolution, where we're trying to pull out tons and tons of data using the AI to quantify all these kinematic trajectories of what they're doing over time, is that then we've got to make sense of this data. So in our pilot cohort, here we're just visualizing a very, very small subset, just 2% of our test run, if you will, right? Where now we're scaling this up to not 4 units, which was what was generated in our test data set, but rather several hundred of these units in which we were recording many mice at a time. So how do we make sense of this massive amount of data? Well, enabled by the low latency and high read and write throughput that our VAST system gives us, we're able to then basically iterate through all of this massive amount of data, do all the AI inferencing, and then pass it through to a probabilistic model that can then characterize the structure, essentially the body language of these animals based on the trajectory. And this is all by using this software called Keypoint MoSeq. It's a model created by our collaborators at Harvard. And it works by essentially just detecting the syllables of body language from the kinematics. And without going into too much detail, suffice to say that one easy way to screen for disease is just to ask, how does the body language change late into the disease versus early? And when we look at an example of some of the syllables that happen less late into the disease, it includes these behaviors like this climbing, something that you expect that would happen less if you have a neurodegenerative disorder. Similarly, we can do this with the entire behavioral repertoire and basically find all of the behavioral biomarkers, all of the little movements and tics and higher order behaviors that are going to correspond to the progression of the disease. And now we can leverage these as a non-invasive, high throughput method for screening through diseases, as well as evaluating whether particular or putative therapeutics might have an effect on the disease and its progression. And finally, since I'm at a NASA conference right now, I thought it'd be thematic to mention that one of the other projects that we have ongoing that leverages this technology is one in which we're doing the exact same thing, but with mice in space, where the goal here is to characterize the space-related change in health and other phenotypes that might give us more information and tell us about how what the risks are and what the biology is of long-term space travel and exposure to space radiation. Okay, so with that, just want to thank lots and lots of folks who made this work possible, our funding sources, and you guys for listening. So thank you. I think you're muted, Jeff.
I said, oh my God, I have so many questions. So first of all, were those mice floating in space, like anti-gravity mice?
They are in the International Space Station in low Earth orbit.
Oh my God. Okay, that's crazy. Okay, so I want to ask a few kind of science questions, and then we'll get into systems.
Mhm.
Let's go kind of most recent to least recent in terms of the topic. So phenotyping. If I was to make use of this, like, so let's say there's human application for finding things like Lou Gehrig's disease. How would I apply that, right? So you would need to have like a camera in somebody's house in order to determine behaviors. At some point, like your Nest will become, or something like your Nest, will become a medical instrument that can be used for early disease detection. Is that kind of a fair way to think about it?
I think so, yeah. I mean, ultimately, that's where we want to go. We want to go from the more controlled settings to the less controlled settings. But eventually, there's really no limitations to this technology in terms of where it's being deployed. So as soon as we can get it into the home, I think it's going to be really, really transformative.
Wow. That's there's all sorts of like HIPAA considerations when you go that route, but the alternative is like sitting in a big white room at a hospital and just kind of like interacting with the universe for, I don't know, an hour while I'm being watched or something like that. Doesn't seem like it's that practical. So
It
sounds
does
like
not.
you're,
And it's not very
yeah.
equitable either, right? Like not everybody can, has the benefit to have that kind of care, especially when we're talking about new medical technologies. I'll say that it's the same philosophy that we apply to the animals, right? Like we could put them in 1,000 different boxes and ask them to learn how to press levers in the right sequences to get the right reward. But instead, the tack that we take is, no, let's just watch them in their, as close as possible to natural habitat. We're not going to touch them. We're not going to ask them to do anything weird. We're just going to let them, roam freely and exploit the richness of their natural behavioral repertoire. And I think the same thing would apply to the human setting, right? I mean, kids these days are essentially on TikTok all the time. They're recording, the same dance, the same sea shanty or whatever it is that's popular this week, right?
What? Is
Every
that a thing?
That's a young thing. Yeah, sorry, Jeff.
What does that mean?
Okay, I heard about it in a podcast. It's a thing kids are into. I think that in the future, you could imagine essentially doing the equivalent of a TikTok dance, right? Once a day for 30 seconds. And the variability and the change in your movements is going to be something that's going to be a strongly predictive signal of the progression and onset of diseases.
Interesting. Okay, so you were starting to talk about applicability. It sounds like, there's a healthcare app that the doctor sends to you and says, do these 7 motions and we'll determine if you've had some sort of regression or something like that. All powered by deep learning, which obviously you probably do inference in the phone at that point, given how strong some of these edge processors are. What about other applications, right? Have you been approached by surveillance organizations, or have you been approached by Hollywood, for example, to do like mocap augmentation or things like that?
Yeah, we have. The surveillance stuff, I'll say that from Frankly, we draw a lot of inspiration from that literature because, as dystopian as it may be, like there is a lot of money in that has gone to developing these technologies. Usually a little bit more specific to like getting particular types of analytics. Things like, I'll say that probably one of the hottest industries is not necessarily security, but actually retail, right?
Sure.
Yeah, a lot of the big name chains, pay a lot of money to be able to extract from their pre-existing security footage, analytics about where do customers spend their time. Does a particular display at the end of an aisle like really attract more attention and more interaction? But the other one, maybe giving retail its money is sports analytics, right? Where there, I mean, it literally is all about
Sure.
money. Optimize your movements by to shave off a couple of milliseconds. That gives you a complete competitive advantage. Yeah.
Okay, there's a bunch of questions that came in too. Let me get to a few of these if you don't mind, just so that some of these things are very topical right now. Frank asks, how are you tracking accuracy of these, I guess, models that get created?
Yeah, sure. So I mean, I kind of showed the results of lots of different kinds of models, but and I don't go into too much technical detail about it. It's really not how we do like our evals. But suffice to say that we use principal metrics that can capture everything from localization to grouping to identity-based errors. All these have different metrics, and there's different ways to kind of take cross sections across those. But effectively, we just follow the standard best practices of having training, validation, and test splits that are held out, cross validation, and doing all the statistics on it, as well as developing metrics that try to account for things like uncertainty and what is correct when there's a fuzzy area.
But it still has to be pretty manual, right? Somebody, like, you have to have a human in the loop to kind of validate even the kind of the accuracy estimation.
Yeah, which is a challenge, right? Because humans themselves are not very
They're terrible.
Yeah, so that's something that one of our metrics takes explicitly into account. This is something called the object key point similarity score. And effectively what it does is it actually models human uncertainty at the body part wise level, where just to give you a concrete example, if I, let's say that I take the consensus of 100 people trying to click on the key points on a human, as like the ground truth, then if I come in and I click and I'm being sloppy and I'm 1 pixel off in terms of labeling the eye, that's going to get penalized a lot more than if you're 1 pixel off and trying to label, say, like the left hip, like some key point that's like corresponds to a very internal, very fuzzy internal part of the anatomy that might even be covered by clothes and other things, right?
Oh, interesting.
Getting it off by 1 pixel, not as big of a deal as getting getting the eye off by a pixel, where you have the iris and lots of other kind of, it's a very punctual, it's very, very well localizable feature. So we take that into account as well when we're factoring in human ground truth.
Okay, so you're using in some sense at least a weighted statistical model of, some might call it machine learning, to kind of grade the model accuracy that you're creating for deep learning.
Yeah.
Okay, so Frank has one more question. He says, how do you calculate H0 and H1 error? I have no clue what that means. So maybe interpret that for us lay people.
I think it's about hypothesis testing. I'm going to go ahead and punt and just say that we leave a lot of that to our experimental collaborators, let's just say, because a lot of that really just comes down to study design. If you have like a multi-arm study that maybe won't even be as necessarily applicable, but suffice to say that there are new statistical and probabilistic methods that need to be developed to account for the fact that we have just such massive amounts of data. And for that, we're really taking a close look at how people deal with this in genomics, where they've had to really grapple with this for much longer than we have. Because in the field of behavior, I mean, the ends, like the sample sizes have been on the order of like, dozens for a really strong study. But now we're talking about generating billions of data points, right, in a single session. So it's it's a different ball game, and we do have to kind of correct for some of these things, and some of it just hasn't been developed thoroughly enough.
So we're gonna talk about algae in a moment. Well, no, sorry, we're gonna talk about brains in a moment. But Cappy asked, and this is probably a good segue to the VAST thing. So what's the average data set size that you deal with when you're doing model development, model training, and evaluation? And do you find there's a limit to the size of the data set that makes the training of the model effective versus, you kind of have a law of diminishing returns?
Yeah, that's a great question, a very timely one at that, right? So I really glossed over that during the talk, but effectively that is something that we care about a lot, and that we've essentially really motivated some of our initial work in this domain to begin with. And this is what is known as the sample efficiency. In other words, for given a single sample or labeled image, how well do you do in terms of how your model can perform, right? And here's a slide that I skipped earlier, which is, when we measure the accuracy as a function of the number of images that we've labeled, or in other words, the data set size, even with as few as 10 labeled images, with our lightweight models, you can get away, you get pretty reasonable accuracies. The outer circle corresponds to where 90% of the predictions fall relative to the ground truth. And while it's really bad for some key points, it's really good for others. But by the time that we get to around 250 labeled images, right, it's still a very modest data set size, the accuracy gets to be pretty much perfect. Now we've reproduced that particular experiment a bunch of times in a bunch of different data sets that have different properties, and this generally tends to hold. But what we've been noticing, like very recently, is in taking a look at how all this whole hype around generative AI is really kind of grounded in the idea of having foundation models. We start to reevaluate some of those curves, because really what we would see was that previously by the time we get to a couple thousand images, basically there's like fully diminishing returns, like very, very diminishing returns, to a point where it's doesn't even make sense to keep going, where it will take like hundreds of hours or more additional labor to get 1% better. Sorry, yeah,
But you have an exponential curve as you get closer to 100%, right? And so if I'm doing things like, tracking fruit flies trying to have sex with each other, then, maybe I have a different standard of what is considered to be acceptable accuracy versus if I'm installing a camera in somebody's house that they need to do like 15 different things for so that you have an FDA approved model. Like I could imagine exponents more computing and data infrastructure for that type of application.
Yeah, so I mean, I would say that in general, actually, the scientific applications tend to have higher, have lower tolerances really to these kinds of errors, because we're really looking at biology down at the millisecond and millimeter scale. But nonetheless, the point still applies, right? There are going to be cases in which we need a lot more precision. And it turns out that you're absolutely right. I don't have a good slide immediately ready here to show you, but basically we've been putting this hypothesis to the test because, like I said, we made our software so easy to use that, unbeknownst to us, lots of users we came to learn ended up labeling tens of thousands of images, even though we ourselves had never gone to more than a couple thousand. And so taking advantage of that and seeing how well their models have been performing, we kind of began to revisit this hypothesis of what happens when you have really large data sets, and revisiting some of the scaling laws as it pertains to model size. And what we find is that even with our relatively modestly sized models, now we're talking like models with 10 million parameters, not like 70 billion, like some of the typical large language models, but 10 million, we still do not see the limit of where it starts to get better. What we were doing wrong before is that we weren't measuring the accuracy correctly. Instead, what we're now doing is we're just measuring like the long tail of the accuracy. Like the average accuracy, it plateaus out within a few hundred images. But when we look at just the 99th percentile of our accuracy, that keeps going down even all the way up to tens of thousands of images. So we're really, focusing on like that long tail now, as well as generalizability.
A lot of long tail work as you're looking at mice tails. Sorry. Okay, so data, right? Okay, let's talk a little bit about VAST then. Is it fair to say that you are not a classically trained systems person? Yes or no?
Yeah, that's correct. Yeah, in fact, I was very glad
Still
to
are
not.
not.
Yeah.
How
I feel
did
like,
how did you get introduced to Vast, or how did you meet our team? Just like
yeah. So, I mean, I think it's a pretty common story in academia. Um, you know, uh, Salk is a very well-known, prestigious biological research institute with some very famous AI scientists, but for the most part, an emphasis on biological research, right? And I kind of came in being at this sort of intersection where I'm doing all computational work, all AI work, but just pointed at biology. And one of my asks when I got the job was just, I needed my compute resources to be able to do it. And, not being a large scale high performance computing type of research institute, they were like, okay, sure, we'll invest in a bunch of GPUs. How hard could it be? And when I started, and we started actually slotting stuff into our data center, and we began to realize like, oh, okay, yeah, we're really not set up for modern research computing infrastructure. We kind of had to take a step back and realize, okay, well, if we're going to scale past like a few dozen GPUs, then we need to think about how does the data, all the data sources connect to it. Make sure that our networking can keep up with the throughput and latency demands of doing machine learning workflows, and more importantly, right, like how can I just get 10 terabytes? This whole conversation started as it does with many academics who are starting off in this whole sphere of just like, hey, I have a new project. I need 10 terabytes or I need 100 terabytes. What do I do here? And the fact that I started getting launched into a conversation, they're like, well, okay, what are all your needs and what are all the, what kinds of kind of replication strategy are you going for, and what is the connectivity protocols and what are they going to talk to, and what are the economics of the particular data workflows that you're going to have, and how we're going to set up all the tiers. The complexity was like, not just mind-boggling, but just bewildering that I even think about it.
Had you ever run a GPU server or a network attached storage system before this?
No, definitely not.
Well, you're a quick study then. Yeah, you got it pretty quickly, I would say.
I think that scientists tend to be this way, particularly neuroscientists. We
That's
arguably are
true.
disciplined, but we just have to do so many different things. I had to learn photonics. I had to learn, 3D printing and fabrication. I had to learn fly brain surgery, as well as, be a naturalist and watch a lot of flies have sex, just to try to understand a little part of their brain. But at the same time, I also had to learn all the AI so I can analyze their body language. So we're kind of used to this whole notion of just having to jump and just learn a whole new technical skill set. But this is one, enterprise storage definitely gave fly brain surgery a run for its money.
Oh my gosh. So the takeaway here is if you want to get smarter, practice by watching a bunch of flies make love. Okay, so
Our worst ways.
yeah, professionally, of course. So wait, you didn't say how you met Vast yet. I don't know the answer to that. So
Oh yeah, sorry. I was bewildered by our, essentially going through our whole NetApp purchasing workflow and requisition and just going back and forth with IT on it. And then that's when I started kind of looking around because I wanted to see, investing all this money on our GPUs makes sense to, kind of reevaluate the rest of the stack. And that's where, we got to seriously thinking about storage, and a lot of it just emerged out of the fact that I needed a little bit more storage. I asked around, there's not a good standard solution. And the refurbished NetApp that that I was sharing with several other people was just really not cutting it. We were spending more time on troubleshooting and working around it than just, it being a layer that we just don't think about. And so when we heard from our partners at Cambridge Computers about Vast, and it was kind of like the hot new thing, and that we should definitely check it out. Diligence. That's when we saw like, yeah, this seems like it would really fit the bill. It simplifies a lot of things, and it basically would fulfill any of the needs that we could possibly foresee.
and how much it just How much administration time does it take from you to kind of babysit this thing?
Approximately 0. Yeah, I mean, it is.
That's
We've
cool.
been, yeah,
I've never heard of any issues with Salk, so I
no,
that,
it
that's
just,
cool.
it was all white glove. You guys, they came in, they installed everything, and you know, it was pretty much turnkey from day one. We had some hiccups with like our authentication, but then that turned out to be on our end anyways in our SSO and Active Directory integrations. But once we sorted that out, and Vast was very helpful in just pointing out where what we were doing wrong, it pretty much just worked right off the bat, and it's been the case for over a year now.
That's cool. I like to say sometimes that we try to take the computer science out of doing science, and I think that maybe you're living that. Okay. You also mentioned Run AI. We just made an announcement with them today about like a kind of an integrated, unified stack that we're building with them as they start to do things like help users understand their data that they need to do, like let's say load into a job. We're going to start listening to that as part of this geo-distributed namespace model that we're building towards. So so that's cool that you're also run any kind of commentary on that, because it seems to be kind of like an equally hot property in the deep learning space.
Yeah, definitely. I mean, orchestration is another one that I think I had thought about a lot more. So I should say that, I before Salk, I was doing my PhD at Princeton, and they had a really top-notch research computing department and group and infrastructure. And so that's where this whole idea of like, oh, I can just email somebody and I get 100 terabytes like instantly, came from. I now realize it's because there's like, many sysadmins that made that
Right.
magic happen behind the scenes at a scale that's just not feasible at a place like Salk that's like 10 times smaller or something. So there we had, we ran everything on Slurm as our HPC orchestrator. And, it was essentially like, it felt like, I mean, I still remember my first year there. It felt like we were just, reciting incantations out of like a mystic spell book to get the right allocations, right? And it's
Yeah.
like, you get you just request 1 megabyte less and your job will queue 6 hours ahead of the other one, and all these little crazy hacks and all this. I appreciate where the orchestration system came from, but I just didn't feel like it made sense for the GPU for GPU times. Where for us, the biggest selling point of Run AI really was GPU fractionalization. The fact that I can request a job with 10% of a GPU if I'm just doing development, or if I just want to have like a sort of virtual workstation. There was that, and the fact that everything is container based, right? That makes it a lot more practical to then move a lot of our workflows to the cloud, for example, if all of our core stuff is already in Docker containers. And so between those two and the fact that our GPU servers are going to sit right next to Vast, and we can just have everything talk to each other really easily. I mean, it just takes a lot of the hassle out of the like our daily workflows for research.
Okay, so let's talk about mega projects. Salk has been in the news by getting some really large grants. Can you explain the Brain project really quickly? Because I have a question about that I didn't ask you, but it's pretty obvious coming from a guy with deep storage roots when you start talking about mapping the brain. Go ahead, you explain it.
Right. Yeah. So so the NIH Brain Initiative is it's essentially part of the NIH, a funding agency whose goal is to develop new neuroscience related technologies. And one of its flagship projects is this BICAN project, which Salk just received this past year, a massive like 80 something million award, maybe 100 million something total award, to essentially leverage sequencing technologies to sequence every cell in the human brain. And this is follow-up work that they had done to sequence every cell in the mouse brain, which, you know, it's something, you know, it's to give you a sense, a point of reference.
Every
you know,
cell.
Yeah, every cell.
How many? Do you know how many cells are in the human brain?
Yeah, between 80 and 100 billion, right?
Oh my gosh.
So it's a lot. It's a lot of data.
Well, that's the question. How much storage is that?
That is beyond my realm of knowledge, unfortunately. So I don't really do much bioinformatics or sequencing, but I do know that in talking to my colleagues at Salk who are directly working on this project, it is something so daunting that they haven't really even figured out, essentially. I think the NIH is requiring them to put all of it on the cloud. Maybe not the primary data, but pretty close to it. And I think they're just going to figure it out from there. So it's just at a scale that, you know, there isn't even like a full plan before the data started getting collected. Maybe that's not fair to say, but basically it's something that is going to be an ongoing process to develop infrastructure around.
Do you use a gene sequencer to sequence a cell, or how do you use it?
Yeah, so there's a variety of different methods that people employ to sequence stuff at scale, but one that's particularly promising and that I'm 90% sure is being employed in this project is what's called spatial transcriptomics, which is this process in which you essentially sequence things just in situ, in tissue. Effectively, take the brain, you're gonna fix it in some way, slice it up, stick it under a microscope, and essentially run chemical processes through it that will essentially cause different colors of light to pop up like a Christmas tree. Then you can take a movie of, which then you can use to reconstruct the sequences, all without actually having to take out the cell, purify it, and run it through a classic sequencer. And the advantage here, obviously, is that you can do many, many cells all at the same time by just sticking the whole big tissue section under the microscope. So there's a lot of
like a light sheet microscope or something like that.
Yeah, something like that. I think they've kind of adapted similar premises to light sheet.
Okay, so you're talking like billions of cells range. You're talking about hundreds of petabytes, probably.
Yeah, probably. Especially when it comes to the raw data, which is now images rather than, you know, individual reads, which are a lot more compressible.
Yeah.
It's a different beast.
That's wild.
Hey, Devin.
Okay, well,
I was just gonna wrap us up. I think we're doing the same thing, Jeff.
Boone's here to give us the hook.
Thank you, Jeff and Talmo. Thanks everyone for joining. Sorry for cutting you off. But thank you everyone for joining us this afternoon.
Awesome. Thank you.
Okay, and thanks, Talmo. Thanks for joining. This was a nice chat.